to today's episode of Myeloma Crowd Radio. Today's show is unique in that we have 3 guests covering an up-and-coming topic, nanotechnology. For those of you like me who are new to nanotechnology, it's best described as the ability to study very small things, or to see and control individual atoms and molecules. I'm here in St. Louis at the Washingon University Center for Multiple Myeloma Nanotherapy, a new center that will apply nanotechnology to bone cancers and multiple myeloma specifically. They are one of a very select group of researchers who have been awarded a coveted NIH grant to explore the use of nanotechnology in cancer. They were selected because they have enormous institutional resources and support, diverse expertise in myeloma, participation of patient advocates and industry partners, both basic and clinical investigators, and most importantly, nanotechnology expertise. Their goal is to improve myeloma treatment outcomes using these very small atoms and molecules to deliver myeloma drugs with better impact and minimal off-target toxicity, as well as other things that you'll hear about later. With me today are 3 specialists, Dr. Samuel Achillefu, a pioneer in the development of molecular imaging probes and nanomaterials for imaging and the treatment of cancer. He currently leads the translation of innovative imaging technologies and molecular probes from the bench to the bedside. We also have Dr. Ravi Vij, a myeloma expert who is Associate Professor of Medicine at the Washington School of Medicine in the bone marrow transplant section. He leads many clinical trials and has established a large myeloma tissue bank at Washington University with a strong focus on studying the genomics of the disease, which are critical to understanding what causes myeloma and how to cure myeloma. We also have with us Dr. Monica Shokin, an expert in imaging technology like MRIs and PET-CT scans, and the development of small macromolecular agents that will improve the accuracy of these imaging tests. Her goal is to find myeloma cells using imaging technology no matter where they reside in the body, no matter how young or old the cells are, and no matter how much they like to hide in the bone marrow niche. I welcome you all. Dr. Achillefu, let's start by you helping us understand how a technology that has been used in chemistry, physics, and engineering is now coming to cancer, and how you're doing that with your projects at Washington University.
So this is one of 6 centers funded by the NCI to introduce nanotechnology into cancer research. And the purpose is to bring a multidisciplinary team of investigators that can bring their expertise to solving a common problem. And of all the 6 centers, in fact, this is the first NCI-funded nanotechnology-based center to look at multiple myeloma. And so, in a nutshell, what we are trying to do is to ask key questions that are unanswered today and take advantage of the power of nanomedicine to apply it into cancer research. So we have projects, 3 projects that are looking at ways of addressing critical issues in multiple myeloma therapy. The first one is looking at a protein, oncogene, that's highly upregulated in multiple myeloma cells. And the problem before now is that people do not know how to deliver inhibitors to where they should work inside cancer cells. So our team members, led by Greg Lanza, discovered a very nice way that nanoparticles will go everywhere using VLA4 discovered and championed by one of our team members here, Monica Shokin, that can latch onto these cancer cells. And then once it's there, it fuses with the membrane in a way that then it empties the content directly into the cells. That way they are not degraded, they go to the target, they only treat the cancer cells and make sure they die.
So you are using nanotechnology to specifically deliver the right treatment to only the cancer cells. What other projects are you working on?
The other project is asking a different questions. What about those cancer cells that relapse all the time, which is what patients worry about? can we prevent relapse? Can we treat them independent of the tumor-resistant phenotype that's there, or treatment-resistant phenotypes? So in that way, we are applying a type of therapy previously is used only for skin diseases. But we discovered a way to take light and a small molecule, a drug, but at a dose that's so low that it will have no effect on normal healthy tissue. When they come together, the radiopharmaceutical that's used to know you're in remission or somebody has cancer relapsing radiopharmaceuticals. If you take those two and bring them together in cancer cells, then they destroy those cancer cells, independent on whether it's resistant or not. And so with that method, the nanotechnology allows us to do a special type of treatment that's never been done before. And that's what we're excited about too. The third project is looking at where cancer cells hide in the bone marrow. Okay, we know that they go in there, the bone marrow niche allows them to sequester themselves in a way that you can find them. And then they relapse at a later time. And when you say you're in remission, doesn't mean necessarily there's nothing there. And so the whole goal is to say, can we find a way to mobilize those cancer cells, kick them out into the bloodstream, send nanoparticles that are very efficient in killing them, to get to them before they go back into hiding place again. And so that project is led by John DiPorzio to help us make sure that indolent cancer cells, the resistant ones hiding in the bone marrows, can be kicked out and then treated. So the whole synergistic event we hope to accomplish here is that with the different therapy methods we have, by combining immunotherapy with light-based therapy, with chemotherapy, the cancer cells have no chance to survive at all. So that's what this is all about, and we are so glad to have leaders like Dr. Vij, who is now giving us guidance into the clinical arenas, access to patients and interacting with them. So we learn every day how to do this much better.
It sounds like those two projects have specific goals. The first is you want to overcome drug resistance, and the second is you want to get rid of leftover myeloma cells that like to hide in the bone marrow by kicking them out into the blood so the myeloma drugs can find them and kill them appropriately. With those goals, maybe you want to back up a little and give us an overview of what nanotechnology is all about. I, for one, am not familiar with it at all, especially in medicine.
Nanotechnology, nanoparticles, just think about the strain of your hair. It's like 1,000 times smaller in diameter. So basically you can't see it with your naked eyes. But think about it like association of atoms. And they put them together in a way that they create these nice pockets, surface areas that allows us to add a lot of materials into them. And so nanoparticles are very tiny, tiny things we can't see, but they can do so much work, 10 times better, 100 times better than single molecules. So an example is to say, if you are looking at increasing new aeroplanes today, they are beginning to make them based on nanomaterials. And the essence of that is that you can pack so many tiny things with different surface areas within a small volume of tissues. And for us in medicine, the advantage of the nano is that it's small, but we can construct it in such a way that we introduce multiple things at the same time. We can introduce drugs that can kill cancer. We can introduce imaging agents that can allow us to see where they are going. We can introduce monitoring device that allows us to know what's happening over time. So the nano platform is a new way that people now are looking into as a way of tackling these resistant tumor types that before now single molecule will not get there. So a final illustration is thinking of soldiers that march in a straight line and they are walking into war. Of course, then you see them and take them out in series. But imagine putting all those 1,000 battalion soldiers into a small place. all pointing their guns to the same person, all of a sudden there's really mass in number to get to their target by shooting from different directions but getting to the same target. And that's what nanoparticle does for us. It amplifies the effect that you can have and prevent escape of cancer cells down the line.
So when I think about small molecule medication, I think about Velcade or other myeloma drugs. Is that what you mean? I think I may be confused when you're talking about small molecules.
Nanoparticles, think about molecules, okay, drugs that you are taking. Each time you take a pill, you have thousands of the same molecule inside the pill, okay? And they formulate it into a tablet and give it to you to take. And in formulating it, what's being done right now is that they use something like chalk to put them together, and you take that, it dissolves, it does what it wants to do. We are asking a different question altogether. Why don't we take that whole big pill you are taking, reduce it 1,000,000 times smaller, that you won't even see it? Now, we can put 1,000,000 times more in that tube you are going to take to have efficient delivery into your body. And we then make them to be released over a longer period of time in the body. So the nanomaterials are like taking so many small molecules, put them together in such a way that you package them so well that they stick together but only get released to do the work as you want them to go through slow release. There's another type. Another type is when you take some materials that themselves can become drugs. And when you put them together again, the advantage you have is that when they come in contact with tumor cells, they can now release their therapeutic effect right there. So think of nanoparticles not as a machine that's different or a big device. It's the same type of drug you are taking, except that now they are packaged differently to go to a given area and not randomly anymore, and then have effect selectively where you want them to have that effect. So there will be no need for giving long IV introduction of chemo drugs for minutes, or I don't know how long you spend doing that, but more, you can just give a little bit amount of this and you still have more than 100 times efficiency than what you would normally have done. So they are still molecules. They are still like small drugs, but now packaged in a way that they only have an effect in the target area.
So you're saying that using this technology can make the medications maybe 100 times more effective and significantly more specific, only hitting the cancerous cells and not the healthy cells. That is completely amazing. Now in immunotherapy, these approaches target certain proteins like CS1 or BCMA or other targets on the surface of the myeloma cells. Does nanotechnology work in the same way? So
we have two questions. There are two issues there. One is targeting, the other one is therapy effect. For us to target, we are looking at a new agent altogether, which is really not part of the center right now, but we hope to bring it in, that can go to different types of cancer. It doesn't matter what type it is. So we're excited about that. That's coming. What we have now is we are looking at treatments. You asked the question about light. I talked about light therapy and all the rest. Let me explain that in a simple way. Right now, there's a type of therapy that's known as photodynamic therapy, and that involves the use of some drugs that are sensitive to light. And if you apply them to the skin, you allow them to absorb in the area that's injured, then you shine light onto that skin of a given wavelength band of light, and then it generates reactive oxygen species inside there that then allows those cells to die. Okay? It's been used in the hospitals to treat a lot of surface lesions. The problem with it is that for multiple myeloma, it's not on the surface. It's spread everywhere. Where will you apply that? Where will you shine the light? And so what this project has done is that we found out that radiopharmaceuticals like FDG, that a lot of patients take, they undergo that all the time, PET scans, the molecule that helps them see the cancer can actually give out light. And so what if we then make the tumor cells take up this material and give out light, so we no longer need to shine the light from outside? The light now will come from inside the cancer cells themselves.
So most patients are familiar with the contrast they get, either with a PET CT scan or sometimes an MRI can be done with contrast. These contrasts take the radioactive glucose and then light up the scan so doctors can see where the myeloma still is in the body and if it's progressing or not. You're saying that these contrast agents can actually shine light on the cancer cells so that the drugs can target them specifically, right?
Yeah, but then we take that light, when we deliver small drugs drugs that naturally will not be toxic, but only when they see that light from cancer cells, then they generate this reactive oxygen that allows you to kill them selectively. So we've taken away that limitation that you need to bring the light to find where the cancer is, shine it and treat it, to the point where we are saying, we don't care where the cancer cells are anymore. We will find them. They are going to take up these radiopharmaceuticals. We are going to deliver these nice drugs that are light sensitive, and the only place they will come together to become toxic is inside cancer cells. So now the light is coming from within instead of from outside, and the drug is having an effect within instead of in the whole vascular system. So that's the light-based therapy we are having.
That is completely amazing. How exactly does it work to kill the myeloma cells?
The good thing then about it, when you think it's not dependent on the type of this multiple myeloma and the rest, is that the way it kills cancer cells is, there are multiple ways. First of all, it goes through this reactive oxygen that makes them toxic and allows us to then start killing them or stress the cells. In that process, some cells will die, but some that are not dying will express antigens on their surface that then allows immune cells to come in and take care of them down the line. So you have immediate response and downstream response where your immune system will kick in to now remove the remaining part of it. So it's not, when we say it's independent of the genotype or the prototype, that's what we mean by that, that we can get to it, we can stimulate it, we can kill it directly, we can disturb the blood vessels that feed into there through this single therapeutic approach. So that's an innovative aspect we are looking at right now.
There are some myeloma researchers I've spoken with who say there are precursor cells or myeloma stem cells that are the early cells and maybe one reason that myeloma keeps coming back, that they can hide and then develop into plasma cells later, and that they might be different than the more mature plasma cells. So that's one question. Can this technology kill those early cells?
So that's actually a very good question, because how do you know the cancer stem cells or the precursor cells that become cancerous down the line, if they will actually do that? Do you want to start treating all of them in anticipation they will transform into cancer or not? What about healthy people? Healthy in the sense that they have not manifested the disease yet, and that may never get the disease down the line because our immune system may be able to walk through that or not. So the goal for us, which is another major area of research we hope to get the funding to do that, is that there's a new molecule that we believe is telling us which of those cells will become cancerous and which ones will not. And we've used it in pancreatic cancer model, where we have the precursor cancer cells, and then it's pointing to us, these are the ones that will become cancerous, these are the ones that will not. we now like to take it to breast cancer, multiple myeloma, and all the rest to ask that question: can we predict which population of cells that are suspicious will become cancerous and which ones may not be? And the fear is always that of overtreatment. If you start going after everything that's suspicious, then the patient may die of overtreatment, and Dr. Vich will explain that better to you. But those are the issues we consider as we move down the line to optimize the therapy in such a way that we are getting after those that will relapse down the line. The way we are proposing to do it, and this is far down the line, and Dr. Vich will be helping us with that later on, is the preventive care that we hope to establish through this therapy, where a patient that is in remission can go once a year. It can be in outpatient or in primary care and just have a dose of this treatment that makes sure if there's anything coming up down the line, we can as well just get rid of them before they develop into anything significant down the way. So that way you are not over-treating a patient, anticipating something bad is going to happen, but instead you are preventing something bad from happening by just having an annual visit to the doctor and use that as preventive care. That's the approach we hope to establish at the end of these studies with this center to move forward.
So your goal is to prevent relapse using a once-a-year, quote, maintenance therapy. That would be fabulous.
And for the high-risk population, one of the suggestions is that, well, if it works the way we hope it's going to work, then those in high-risk population can always go ahead and do that. The advantage being that both the drug and the radiopharmaceuticals, if we can give them at the sub-lethal dose, sub-therapeutic dose, then the worries that you will have by using current preventive care methods may not be an issue anymore. And because this therapy does not induce resistance by the cells, you will not see cancer cell resistance due to photodynamic therapy and the rest, then repeat treatment is not going to be problematic. You are looking at the new onset of the disease that's coming from somewhere, and they will still be able to respond. But the treatment, even if it's in a preventive care mode, is not going to stimulate therapy resistance down the line. So that's our hope.
That would be amazing. You mentioned pancreatic and breast cancer. Has this been used already in other solid tumor cancers?
Yeah, we may be fortunate actually to get one of the biggest awards in breast cancer to look at that using this approach as well, where we are looking at the types of cancer, especially the spread of cancer, metastatic breast cancer. That's an area we are excited about, looking at the indolent cells that do not really respond well to treatment and predicting which patients will, DCIS will now go on to become real malignant tumors down the line versus those that will not progress and be able to eradicate them that way. So we're excited to look at it because sometimes we see cancer as different types, and we address them there, but at times when it comes to treatment, that's looking at not the current molecular pathway people are using, we can then expand it. If we are successful in multiple myeloma, Dr. Vich may now be calling his colleagues in other areas and saying, do you want to try this therapy in your case or not? Because we can still deliver them and have similar effect. So this center is going to set the stage, the platform, that we now open the door for us to start looking at different types of malignancies that are around today. But we are starting it, the first one in the country that's looking at multiple myeloma as an entity to disease to take care of.
We are so excited that you chose multiple myeloma. How long has this therapy been around? You're the first center to do this in myeloma and the recipient of this NIH grant. How did you get involved with that?
Oh, so officially our center started That was last September. So we are not even up to a year yet. But we've made good progress thanks to everybody here. The way we started was that the NIH required us. They're looking for centers of excellence in nanotechnology that we apply that knowledge to cancer therapy. And so we're looking at, we say, well, we should be one of those centers. It was highly competitive. They already had 9, 10 centers before now, and they are reducing everything to only 6. And the question became, how do we do it? We looked at different types of cancer, and we're looking at solid tumors and blood cancers and everything. We have the technologies that can go through all of them. But the question became, where do we have a niche? If an area we can really focus on and make an impact. And fortunately for us, there was Michael Thomason, Monica Shokin, all of who were working on multiple myeloma. And it turns out that Dr. Ravi Vij has this huge patient population he treats all the time here. So we have the clinical center of excellence for that. We have top-notch researchers that can do multiple myeloma. We have technologies that can be applied to multiple myeloma. And so we asked ourselves, what if we put those 3 pieces together to create a center that will now be looking at it from a holistic point of view? And that's how we came about that.
Dr. Achillefu, thank you so much for joining us today and explaining this program. Your work is completely amazing, and we just hope you keep going. Dr. Vij, you see myeloma patients all day long. In the context of myeloma, how do you see this being used?
So nanotechnology is obviously disease agnostic. It is just a method of drug delivery, and it's even wider than that. Nanotechnology, as you've heard from Sam, can be used in wide industrial applications, things like that. So as far as where we stand with it in terms of patient application, we're still probably, at least a couple of years off. You have to test this technology in preclinical models first. Then you have to do preclinical work, which is, going to form the basis of an IND that will then be submitted to the FDA, wherein they will permit what are going to be, sort of first in human trials. And like any drug, it will go through phase 1, phase 2, phase 3 development. So I think that not many people realize, in some ways, one of the drugs that is out there, Doxil, is also based on nanospheres in encapsulated Doxil, and that is a form of nanotechnology. But the kind of nanoparticles being explored here are totally novel and much better in the preclinical realm to deliver the drugs and do some of the very fancy things, as you saw Sam say, about being activated by light, being targeting things that are very specific to the cancer cell, whereas Doxil is just going to be a drug that is encapsulated in these liposomes that are not really truly specific for a cancer cell. So it is using nanotechnology to be much more targeted to the cancer cell. And in terms of when we bring it to patients, it will obviously be something that we will have to tread very carefully, and because this is absolutely new technology, and it is something that we hope that will be a true pharmaceutical product one day, but I think we're probably several years away from it being a commercially available product.
What I hear you saying is it has to go through the regular process of clinical trials. So why myeloma? What makes myeloma an attractive target?
I think that, as I say, the technology is actually disease agnostic and cancer agnostic. I think the way it has come about, as Sam told you, is because we had a critical mass of researchers, people who had research background in myeloma. They had mouse models, they had petri dishes full of myeloma cells that they had been working with already. They had produced data independent of nanotechnology researchers. And then those two groups met, and based on what they saw, the nanotechnology group had already been happening. They felt that it was a prime area because of the strength of the scientists that are already engaged in the basic science research in the realm of myeloma to then use technology that they were developing to come and give patients hope in myeloma.
It's not often you have everything you need at one center to go after a specific disease with a full team and a 360-degree view of the disease from the research side to the clinician side with patients.
You have to realize a lot of the people in the nanotechnology world that are participating in the development here are by training cardiologists. Some of them are individuals that have a background in radiotherapy and imaging. There are others that are actually engineers. They work in the School of Bioengineering at Washington University. So it's a multidisciplinary approach. The synthesis of these nanoparticles is done by people who have a background in chemistry and engineering. So they were obviously were already working independently on creating these materials. So it was a good synergy that came about, and some of the other things that played a role, as Sam said, is the demonstration to the MCI that we do have a critical mass of patients where these technology can subsequently be developed, and the transition could be made from the laboratory to the clinic. So I think that it is, we're very glad that it is myeloma, but could it have been breast cancer or lung cancer or colon cancer? Sure. The technology could be applied to any cancer, but you need to have researchers that are like-minded with a focus, and that's how it came about. And I'm very happy. I'm a clinician, and I will hopefully be able to utilize this for the good of patients. But I have to rely on my basic science researchers like Sam and Monica here to be able to, develop the technology to be able to make that transition.
Well, I congratulate you on the depth you have here and the coordinated effort. What you're doing is not an easy task.
I think that there are very few centers that have the ability to bring together a multidisciplinary stream in any cancer type. And I think that Washington University, we are fortunate that we have some of the leading researchers in various fields, and each is a world-renowned figure in their own area. And it is breaking down the barriers. It is, people work in silos, and you don't know that a technology being developed literally in the next room could be used by you, who is working on a totally different area, to help patients. And I think that that is the challenge of science, to often break the barriers. Often the technology exists. It is just that you don't know the person in the next room can utilize your technology and take it to the next level. So yes, I think that there are quite a few centers that have the capability to do so, and there are other centers that are working from a multidisciplinary approach. But yes, there are probably in the United States perhaps half a dozen, definitely less than 10, that have a programmatic approach wherein you have the critical mass of basic scientists and scientists in different areas that can come together to do it. Some people do the clinical piece very well, others do the basic science piece, but lack the clinical ability to translate it to the clinic. We're fortunate that we seem to have all the pieces here, and we hope to be able to utilize it to make it happen, to make the transition.
So in today's world in myeloma treatment, we always hear about combination therapies. A stem cell transplant added to a proteasome inhibitor or an IMiD, a steroid, and maybe an immunotherapy. How do you see this being incorporated, or is the hope that it replaces everything?
Well, I think we'll have to see how it goes. Obviously, we are learning more and more about the genetics and the genomics of myeloma. We are learning that these cells have not got these cancers don't have a homogeneous group of mutations even within the same individual, and that at different times different groups of cancer cells with certain mutations predominate. And that has led to the realization that we need to give our drugs in combination so that you can target different subsets of cells within the tumor. But the fact is that there is also another countervailing paradigm that's evolving with immunotherapy, where it is felt that you may not have to perhaps target individual cell mutations, that you can eliminate cancer by just having the body's immune system attack the cancer. And so I think where this battle, or this hopefully in the future will be synergy, leads to is an open question. Will we need to develop drugs targeted to individual mutations, combine them, and kill away individual clones of cells within the tumor to be successful? Or could we have an approach where you can actually target the cell independent of its genetic makeup? And as I said, nanotechnology is a mode of delivery, and it could possibly have applications in both those paradigms. You could conceivably have Velcade attached to a nanoparticle and Revlimid attached to a nanoparticle that will be much better, less toxic, and be more able to kill the cancer cell better. On the other hand, you can have perhaps the nanotechnology used to deliver a molecule that will kill the cell based on either an immune approach or a cell surface molecule that is present on all those cells irrespective of its genetic makeup.
I think I'm beginning to understand that this is a drug delivery method, not another type of drug or treatment in and of itself.
It is a drug delivery method. It is. It's not only just a drug delivery method. Nanotechnology is useful in a lot of areas of, as I said, industry. It has so many wide applications just outside of medicine. It is used outside of medicine even. These are just using very small molecules, as Sam was saying, 1,000 times smaller than the breadth of a hair, to then have a structure that can accommodate drug molecules for delivery in a very concentrated manner.
You could even use it to deliver melphalan or something like that.
Very true. You could possibly attach melphalan to a nanoparticle for making it much more efficient and less toxic. But we also hope that we'll not only use this technology to deliver drugs that are out there and people are already working with preclinically, but also to deliver drugs that are synthesized and are formulated here, which would take it to the next level. You have the delivery method and you have the novel molecule, like what Sam was talking about, the light-activated system. That is something that is being explored here. It is something that has been developed here. So you have a nanoparticle developed here, delivering a drug method or a postulated new method of killing cancer cells developed here being combined.
Dr. Vij, thank you for giving us a myeloma perspective. One thing both you and Dr. Achilefu talked about is using imaging like PET-CT, a PET-CT type of technology, to actually deliver targeted drugs. Dr. Shokin, you're an expert in imaging. How did you get involved in this and in nanotechnology?
So I have a PhD in chemistry, and I've been trained as a chemist from Washingon University in St. Louis itself, and then I had a wonderful opportunity to do my postdoctoral work here at the medical school. interestingly, under the Program of Excellence in Nanotechnology. So during my early years of postdoctoral work, I was exposed to nanotechnology and nanomedicine in particular. And that's where I developed interest in developing nanoparticle-based imaging agents. So I found my passion into designing radiopharmaceuticals for cancer research and cardiovascular research. So the chemist, we could go to the drawing board and think of molecules that could target cancer cells more specifically and use as an imaging agent. So about molecular imaging in the preclinical models and clinical imaging, it has the advantage of giving a whole body picture in real time. Okay, so we know that a person has a disease through blood tests, urine tests, but molecular imaging and imaging in general gives us the power to see where the disease is located. Is it spread, how widely it is spread, and which lesions are more active and which lesions are less active and benign and could be left untreated. And so I started working with nanoparticles in terms of designs of drug delivery. And initially I started working with breast cancer targeted nanoparticles, and I worked with my collaborator Catherine Weilbacher, who is a world-renowned breast cancer clinician here at Washingon University. And so we looked at targeting breast cancer, and then I had the opportunity to work with the myeloma group here at Washington University with Michael Thomason, Ravi Vij, and John DiPergio. And what intrigued me at that time, as I was starting my own independent research group, was that at that time in 2012, there were no targeted molecular imaging agents for multiple myeloma or leukemias in general.
So most patients are familiar with the PET scan, where they have this glucose radioactive material, and the glucose goes to the active myeloma cells, and that's what shows up on their scan.
FDG PET has been the workhorse, the gold standard for oncologic imaging, and to a very good value. So it has served the patient community and continues to do that in terms of detecting disease and then monitoring therapeutic response. But FDG is looking at one aspect of the cancer proliferation. So cancer cells love glucose to survive and proliferate, and that's where FDG comes in. However, I was interested in looking at how about the lesions in the bone marrow that do not express this receptor, the GLUT1 transporter, which is needed for the FDG uptake. So can we have something more specific for myeloma cells? Can we learn more about the biology of this, maybe the resident phenotypes, drug-resistant phenotypes of myeloma? So with that motivation, I started designing radiopharmaceuticals for the targeted imaging of myeloma, initially in preclinical animal models. I was fortunate at early on, right after I joined the optical radiology lab under the mentorship of Professor Samachilifu, I was awarded an R01 grant by NCI to develop receptor-targeted imaging agents for multiple myeloma, because even NCI saw the need that in their portfolio of imaging agents, what was missing was a good imaging agent for multiple myeloma. The challenge there was, well, the disease is in the blood, in leukemias, how do you see focal points? But myeloma, is one of the characteristics of myeloma is that you have multiple focal lesions within the bones. So there's bone imaging, and then there can be the functional imaging to look at the proliferating cells, and can we target those cells and help in the diagnosis of, in the early diagnosis and more efficient diagnosis of myeloma patients. There is a subset of myeloma population where which are oligosecretory. So those myelomas are missed by the traditional test of urine tests or blood tests. And Ravi Vij always used to tell me that's the population which really needs imaging, because we cannot see it by our routine tests, and that's a false negative. And so imaging can be part of that. And
Now, these are patients who have essentially lost the M spike, right? So they're called non-secretors, or they're no longer making the M protein. So it's tough to test their blood or urine, and you really have to do a biopsy, which everybody wants to avoid. What you're saying is you can use imaging instead.
Then looking at the resistant cells or following therapy, that's something FDG sometimes can have a higher background uptake in the bone marrow. So we want to. So I started working on the agents that can be more specific, bring in more specificity while retaining the sensitivity of PET, which is a positron emission tomography used in oncologic imaging, which gives us a high signal to noise, and it does not have to deal with the barriers of light because it can be easily detected. These are high energy gamma rays that can be detected by the camera around the patient. So with my grant, and now my group is actively working at looking at different molecular imaging modalities to more effectively diagnose, image, and monitor multiple myeloma. And I've been very fortunate to be part of the Center for Multiple Myeloma Nanotherapy, because here I'm working with the different projects to monitor the therapeutic response more effectively, in addition to the SPEP, or the free light chain assay. Can we look at which cells escaped the therapy? Can we identify those and then target those? And as Sam explained, that we need a good source of light. FDG is a fantastic source of light of getting to most of the cells, but can we have something a different, in addition to FDG, a more receptor targeted? In my case, I'm looking at VLF4.
And what is F2G? I think you may have to explain that. Now, this is the contrast we get when we get imaging done?
So basically a glucose molecule. So what chemists realized early on was, this is cancer cells love glucose molecules. So what we did decades ago, and FDG was one of the very first FDA-approved imaging agents used in clinic, was to replace one of the atoms on the glucose molecule with a fluorine, a radioactive fluorine atom. So in what radioactive fluorine, it's a PET radionuclide. So radioactive fluorine gives out positrons. So it's a positron radionuclide. So what it does is that we have changed a regular non-radioactive glucose molecule into a radioactive glucose molecule, which is now, so cancer cells take it up as they would take up glucose. They think it's the same sugar molecule, it's sugar to them. And so once it's taken up by the cancer cells, interestingly, since we have now replaced one of the atoms in the glucose molecule with the fluorine, they don't know how to use it up. It's pretty much trapped in the cancer cell. So the more they take it up, it's just trapped, and then you get a more higher signal to noise. It's a light bulb which is getting brighter and brighter as it's taken up because of the radioactive fluorine 18, which is now part of this molecule. And here at Washingon University, we have a world-class cyclotron facility, so we are leaders in making FDG, a clinical-grade FDG, which we give to patients all the time. Well, no, these are two different things. So in MRI also, the contrast you get is a gadolinium contrast. So MRI is definitely one of the star imaging agents, imaging platform for multiple myeloma patients because of its very high resolution and specifically giving the soft tissue contrast in the bone marrow. So for looking at bone marrow imaging, gadolinium enhanced MR. So that's the contrast agent you get. However, that is not specific. It's basically looking. It's useful for giving. Looking at the different anatomical parts of the cortical bone, trabecular bone, pelvic bone, and also looking at the bone marrow. So when there are tumor cells, the way water diffuses in and out, there's a differential, so you're essentially measuring that change of water diffusion. So MR, magnetic resonance imaging, is basically looking at water molecules, which we have plentiful in us, and so we try to create that contrast by giving gadolinium. So that's one imaging platform. PET is considered more functional platform, where we are looking. So MR gives a great anatomical contrast, and also we are picking up a lot of more functional biomarkers based on MR. PET is very sensitive, and so the cool thing now is that we have more combined systems, a PET MR. So usually if a patient gets a PET CT, so CT scan gives a very nice snapshot of the bone. Okay, so it helps you localize a PET signal. It gives it a location so this is close to, the arm, upper arm, or leg, and so you have a very nice co-registration. But now we are doing here at Washington University. We have I think we were the third one in the whole U.S. to get a clinical PET MR scanner, and we are screening a lot of myeloma patients who are using that. So where we are using the power of MRI and the PET, but all together.
That's fantastic to get them both done at the same time. Very efficient.
Otherwise, you would have to independent scan. So for MR, you'll have one, and you'll be given a gadolinium contrast, and then for PET, you'll be given an FDG PET. So that's a different contrast. They have different half-lives, and you cannot. So your PET is more high energy. It's called. It uses ionizing radiation, but it's given in. The contrast is given in such low doses that it does not cause any harm. So it basically, it's called non-invasive imaging in the sense that it's an innocent bystander taking a picture of the tumor, but not really interfering with the tumor, and that's what we want.
So now you're trying to apply those lessons you've learned about imaging to nanotechnology.
Rather than shooting in the dark, we are giving the clinicians a real-time snapshot of where the lesions are located, okay? And then some lesions would respond, and then you could see by FDG PET or any other more targeted contrast agent. So, and then you can hopefully manage the disease more effectively and identify the responders from the non-responders at an early stage rather than waiting several weeks or months before we realized, a therapeutic change should have taken place much earlier to help the patient. So basically helping with the management in addition to early diagnosis. And also, as part of the center, so the center is looking at effective ways of treating multiple myeloma cells. And part of the center is also using nanoparticles. So like Sam and Ravi mentioned, so nanoparticles are multifunctional molecules. So we can attach different drugs. We can encapsulate different drugs into the core of the nanoparticle and then have more targeting agents around it. And those targeting agents could target, like you mentioned, CS1, VLF4, or CD38 on its surface so that they can chaperone these nanoparticles to where the tumor cells are and have it retained there. And then we can attach imaging agents to these nanoparticles. So in addition to delivering therapy and using light also as part of the therapy, we can image how well our therapy is working, if it's reaching the target. So it's one of so it's pretty much giving it a flashlight to our therapy.
And you're saying, how well is this working?
Exactly. Is it going to, one, to the tumor cells, and two, did we make an impact? Was there a reduction in the lesion in the bone or outside of the bone?
Where do you see this new technology headed?
I believe nanoparticles and nanomedicine in general has evolved a great way. I was involved with nanomedicine almost a decade ago, and I have seen over the years the progress researchers across the world has made. So the technology is ready for the next step, where it can be it's ready to be helping the patients. Now it's more biocompatible, it's more bioavailable in vivo. So that's one of the big effective targeting. And I believe it's going to impact the disease and hopefully help eradicate or manage multiple myeloma more effectively. Yes, as a chemist and as a scientist, I believe in this technology.
Dr. Achalefu, Dr. Vij, and Dr. Shokeen, thank you so much for taking time to talk with us so we can learn about this groundbreaking new approach to treating myeloma. I congratulate you again for your creation and funding by the NIH of this exceptionally innovative program that we hope will help cure this disease. We know we'll be hearing much more about your project in the future, and we look forward to your updates. So thank you for listening to another episode of Myeloma Crowd Radio. Join us for future shows to learn more about the latest in myeloma research and what it means for you.
Can I give you $30 to lean into your decision to start working out and eating better? I'm Carl, co-founder of Body. That's B-O-D-I. And right now, if you sign up for a 1-year subscription to Body, I'll give you an instant $30 discount. That's 59% off. Look, I know it's not easy to get fit and lose weight, especially if you're trying to figure it out by yourself. But we make it simple. Just follow a program for 20 to 30 minutes day by day and lose 5 to 10 pounds a month. We have over 120 programs that have been tested and proven to work, and almost 300,000 5-star reviews in the App Store to prove it. Body also has complete eating plans and thousands of healthy, delicious recipes. So stop guessing and start seeing results with Body. And let me give you a $30 instant discount right now so you save big on the app that CNN_ named Best Fitness App. So don't wait. Sign up for a year of Body and get a $30 instant discount to save 59%. Just go to body.com. That's body with an I.com.
With Lucky Land Slots, you can get lucky just about anywhere.
Dearly beloved, we are gathered here today to... Has anyone seen the bride and groom?
Sorry, we're here. We were getting lucky in the limo and we lost track of time.
No, Lucky Land Casino, with cash prizes that add up quicker than a guest registry.
In that case, I pronounce you lucky.
Play for free at LuckyLandSlots.com. Daily bonuses are waiting. No purchase necessary. Void where prohibited by law. 18+. Terms and conditions apply. See website for details.